Hello viewers, this is Dr. Nirmala from Bishwa's Biochemistry. Today we are going to discuss on an interesting pathway, that is citric acid cycle. The cycle has also other names like Krebs cycle or TCA cycle. So before learning the detailed reactions of the TCA cycle, we will brush up with the points in the introduction, an overview for the pathway and the steps which is taking place, and the pathway with the reactions, and there comes the overall reaction of the pathway and the energetics, and two important pathways which is relevant to the TCA cycle, that is your amphibolic pathway and the anaplerotic reactions. So before seeing the detailed reactions or the steps taking place in the TCA cycle, I would like to tell you a few points regarding the glycolysis and the TCA cycle and its connection with the production of the energy. Coming to the glycolytic pathway, in this pathway, glucose, a 6-carbon molecule, is entering into the reaction which has got 10 steps, and these 10 steps are divided into the preparatory and the payoff phase, and finally it produces 2 molecules of pyruvate. This 6-carbon compound is converted into 2 molecules of 3-carbon compound, that is your pyruvate. And this pyruvate is having 3 different fates. Pyruvate can either be converted into acetyl coenzyme A, and this reaction is a single step reaction which is catalyzed by a multi-enzyme complex, that is your pyruvate dehydrogenase. And this acetyl coenzyme A molecule enters into the cyclic pathway which is your TCA cycle. And in the TCA cycle, we have the production of energy and there is a release of carbon dioxide. So coming to the reactions, we are having 2 different types of reactions. One is aerobic respiration and the other one is your anaerobic respiration. This is based on the amount of oxygen which is present in the cells, and it is decided which type of pathway it has to be taken. So in the presence of oxygen, a series of reaction takes place. That is, your glucose is converted into pyruvate and pyruvate is converted into acetyl coenzyme A. So what happens when there is an imbalance oxygen in the cells? The pyruvate is automatically converted into lactate with the help of the enzyme lactate dehydrogenase. Whereas in yeast and other microorganisms, this 3-carbon compound, your pyruvate, is converted into ethanol with the help of the enzyme alcohol dehydrogenase. So this is what is happening, and it is based on the presence or the absence of oxygen. So we have this acetyl coenzyme A molecule which finally enters into the cyclic pathway, that is the TCA cycle. Coming to the naming of the TCA cycle, it is called as a citric acid cycle because of the first molecule citrate which enters into the reaction. It is also called as a TCA cycle because of the first 2 intermediates, which is a 3 carboxyl groups on the structure. And we have another important interesting name which is given, Krebs cycle, which is named after the discoverer Hans Krebs, who elucidated the pathway and won Nobel Prize for the same in 1953 with Lippmann, who is the father of the ATP cycle. In short, glycolysis is the pathway which is going to convert your glucose molecule into two molecules of pyruvate, and with the help of pyruvate dehydrogenase it is converted into acetyl coenzyme A, and this acetyl coenzyme A enters into the TCA cycle to carry out the cellular respiration, and it harvests energy, produces carbon dioxide, and the energy here is formed in the form of NADH, FADH2 and ATP, and these pass their electrons into the electron transport chain for the ATP production. And coming to the overview of the pathway. In prokaryotic cells, the pathway occurs in the cytoplasm, and in the eukaryotic cells it happens in the mitochondrial matrix, which means the enzymes which are going to catalyze the TCA cycle is present either in the cytoplasm or the mitochondrial matrix, and the cycle is in a closed loop and it operates only in the aerobic condition, which means it is strictly aerobic, takes place in the presence of oxygen. And since it is a closed loop, the last product enters the cycle again. It is a cyclic pathway. The very first product enters as the last one, and again it is repeating, and we have got 8 different steps in the pathway. For one molecule, that is one molecule of acetyl coenzyme A releases 2 carbon dioxide molecule and produces 3 NADH, 1 FADH2 and 1 ATP molecule. And these are the steps which are catalyzed by different enzymes. Coming to the steps, the very first step is condensation, and we have the second step which is A and B, that is dehydration and the hydration process, and we have two oxidative decarboxylation steps which produces carbon dioxide, and there is a substrate level phosphorylation, a dehydrogenation, hydration, another dehydrogenation which releases oxaloacetic acid which enters into the cycle again. Coming to the detailed explanation of each and every reaction which is happening in the TCA cycle. The first reaction is known as condensation because there is a clubbing up of the molecule, that is your oxaloacetic acid and the acetyl coenzyme A, which is derived from the pyruvate. They react in the presence of the enzyme citrate synthase to produce the 3 carbon compound, that is your citrate molecule. And an interesting point in the citrate synthase enzyme is it is the first enzyme in the pathway and it is the first regulating point, which means this particular enzyme is regulated and decides the product from the TCA cycle, which may be needed in the different amounts at different times. The regulation will help the products to be either formed or either it can be controlled at various times. And every time the first enzyme is regulated because it is the substrate for the second enzyme. So the cell can control the amount of resulting product by regulating the first enzyme. Here in this pathway, the regulation point is your citrate synthase, and it can be regulated by ATP, NADH, acyl-CoA and succinyl-CoA. And coming to the second reaction, which has got a hydration as well as a dehydration, both the process, wherein we have a removal of water molecule and addition of water molecule. Citrate is converted into isocitrate, and there is an intermediate product which is nothing but cis aconitate, and both the reactions here are catalyzed by the enzyme aconitase. This aconitase can be inhibited by the inhibitor fluoroacetate. This molecule can inhibit the action of this particular enzyme aconitase. And coming to the third reaction, that is your oxidative decarboxylation. Here, isocitrate is being converted into alpha ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction there is one carbon dioxide which is being released, and there is a conversion of NAD to NADH, the first reaction where you have the energy production. And this enzyme is also a regulating point next to that of our citrate synthase, the very first reaction. And this can also be regulated by the levels of ATP and NADH. And we have the fourth reaction, which is also an oxidative decarboxylation, which catalyzes the conversion of alpha ketoglutarate to succinyl CoA. We have the second carbon dioxide molecule which is being released, and the enzyme which is going to catalyze this particular reaction is nothing but alpha ketoglutarate dehydrogenase. It is a complex. Here we have the production of energy in the form of NADH. Next to the isocitrate dehydrogenase, the alpha ketoglutarate dehydrogenase is also a regulating point which can be regulated by the concentration of the NADH and succinyl CoA. It can also be inhibited by an inhibitor, that is arsenite. The next is the interesting step, that is the substrate level phosphorylation, which is going to yield ATP molecule by the conversion of succinyl CoA to succinate, which is catalyzed by the enzyme succinyl CoA synthetase. There is a conversion of GDP to GTP, and this GTP is converted into ATP by nucleotide diphosphate kinase, and now there is a production of the ATP molecule in this reaction. And the sixth reaction goes with the dehydrogenation, that is conversion of succinate to fumarate, which is catalyzed by the enzyme succinate dehydrogenase, and there is a release of the energy in the form of FADH2. This particular enzyme can be inhibited by malonate. Here we go with the reaction 7, that is hydration. Already we have learned a hydration process in the second step, which is nothing but an inclusion of water molecule. And here in the seventh step, inclusion of water molecule converts fumarate into malate in the presence of the enzyme fumarase. And the last reaction which produces the oxaloacetic acid again, which is going to enter into the reaction, that is conversion of malate to oxaloacetate in the presence of malate dehydrogenase. And here there is a production of again energy in the form of NADH. And here is a pathway which shows all the 8 steps in one particular slide, that is starting from the acetyl coenzyme A, which is entering into the reaction forming citrate, and we have the intermediate, that is the second step, dehydration and hydration club together, which produces isocitrate, and we have alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, and oxaloacetate, which is going to enter into the pathway again. And the orange color circles give out the molecules which are going to produce energy, that is in the form of NADH, FADH2 and GTP. And in the reactions, we have seen two important things, that is the regulatory enzymes. Why do we need these regulatory enzymes in the pathway? These enzymes is done for the pathway whose product may be needed in different amounts and at different times. So these enzymes will decide whether the product has to be formed at that particular time or not. If we do not require the product, these enzymes may be automatically regulated and there isn't any production of the product. So such three enzymes we have seen in the pathways are your citrate synthase, isocitrate dehydrogenase, and the alpha ketoglutarate dehydrogenase. All these three enzymes play an important part in the regulation of the TCA cycle. And we also have the inhibitors, that is the second enzyme, aconitase, which was inhibited by the fluoroacetate, and alpha ketoglutarate dehydrogenase, which can be inhibited by the arsenite inhibitor, and succinate dehydrogenase can be inhibited by malonate. So the TCA cycle pathway can either be regulated at the regulatory enzymes, the regulatory point in the TCA cycle, also can be regulated with the help of the inhibitors. And coming to the overall reaction which is happening in the TCA cycle is, for one molecule of acetyl coenzyme A, we have the formation of energy as 3 molecules of NADH, 1 molecule of FADH2 and 1 molecule of ATP. And this is only for one molecule of acetyl coenzyme A. But we know that 2 molecules of pyruvate is being converted into 2 molecules of acetyl coenzyme A, which means the energetics will be like this. That is, the first column gives you the serial number and the second one is the reaction, and the third column is the number of ATP molecules which is produced for one turn of the TCA cycle. The very first enzyme, isocitrate dehydrogenase, it produces one NADH, which means we are getting 3 molecules of ATP. And the second one is the alpha ketoglutarate dehydrogenase in the reaction which produces one NADH. Now when it comes to the number of ATP molecule, it is 3 ATP molecules. And we have succinyl CoA synthetase which produces GTP, and the GTP is converted into ATP. So we have got one molecule of ATP. And we have one FADH2 which is produced by succinate dehydrogenase and which counts to 2 molecules of ATP. And the last enzyme in the pathway is your malate dehydrogenase, which produces 1 molecule of NADH and counts to 3 molecules of ATP. So total number of ATP molecules which is produced in this cycle of oxidation of one acetyl coenzyme A is 12 molecules. Therefore, for 2 acetyl coenzyme A molecules, number of ATP generated is 24. Now there is an interesting pathway which is to be learned in connection with the TCA cycle, that is our amphibolic pathway, and in the forthcoming slides we will be seeing the anaplerotic reactions. So why do we need this amphibolic pathway and the anaplerotic reactions? TCA cycle is called as an amphibolic pathway because it serves in both catabolic and anabolic process. What do you mean by catabolic process? That is, energy is derived from the oxidation of acetyl coenzyme A. That is, there is a production of carbon dioxide. The oxaloacetate acetyl coenzyme A is finally converted into molecules of energy. And anabolic process, there are several intermediates which act as a precursor in the biosynthetic pathways. That is, there are some regulatory points. The enzymes in the TCA cycle are either regulated by the ATP concentration or NADH, succinyl CoA or acyl CoA, which will uh, just give the pathways to produce products in different amounts at different times. So why do we need this regulatory points? So when the TCA cycle is not to be operated, the TCA cycle intermediates will be acting as a precursor for the biosynthetic pathways. Such a pathway where the TCA cycle intermediates are used as a precursor is the amphibolic pathway. It is also termed as metabolic traffic cycle. And here comes the reactions which are happening in the amphibolic pathway. So the very first enzyme, citrate synthase, if it is being operated, there is an accumulation of the citrate molecule which may be converted into fatty acids and steroids. So when there is an accumulation of citrate in the TCA cycle, the citrate is diverted into the synthesis of other compounds. And we have the alpha ketoglutarate. When this alpha ketoglutarate is accumulated, it is converted into glutamate, an amino acid, and from which three other amino acids, glutamine, proline, and arginine, can be synthesized. And we also have an purine, which is a component of the nucleic acid, being synthesized. And we have succinyl CoA, which is converted into porphyrins and hemes. And we have the last product of the TCA cycle, the oxaloacetate, which is converted into phosphoenol pyruvate, which is converted back into glucose. And the phosphoenol pyruvate is converted into many amino acids. Also, the oxaloacetic acid is converted into aspartate, aspargine, and also forms another nucleic acid component, pyrimidines. So, the TCA cycle as such, the intermediates are diverted to the synthesis of other important compounds. And coming to the anaplerotic reactions. So why do we need this anaplerotic reaction? So when the TCA cycle through the amphibolic pathway, it is being used as a precursor for other biosynthetic products, we need to replenish the TCA cycle intermediates. That is, to bring back these intermediates into the TCA cycle to run the TCA cycle. So such a set of metabolic reactions are called anaplerotic reactions, which helps in replenishing the TCA cycle intermediates. We have four such anaplerotic reactions, which is shown in the table here, and we have the pathway in this. We have amphibolic as well as the anaplerotic reactions, which is being shown. The red color arrow marks are nothing but the anaplerotic reactions, that is replenishing the TCA cycle intermediate. The main part of the TCA cycle is the oxaloacetic acid, which is replenished back with the help of the phosphoenol pyruvate, with the help of the enzyme PEP carboxykinase and PEP carboxylase. And we have the pyruvate, which is being converted into oxaloacetic acid with the help of the pyruvate carboxylase. Oxaloacetic acid is the main substrate for the TCA cycle along with the acetyl coenzyme A to run the TCA cycle. And the next one is the malate, which is being again formed from the pyruvate with the help of the malic enzyme. So we have got four anaplerotic reactions which helps us to take back the intermediates of the TCA cycle and allow the TCA cycle for the energy production. Here we have the overall aerobic respiration that produces the number of ATP molecules. Glycolysis is the first step where glucose is being converted into two molecules of pyruvate. As we discussed in the first slide in the introduction, it has got 10 different steps, and we have a preparatory phase and a payoff phase, and which is going to give us 8 molecules of ATP in the reactions. And the next important reaction is the pyruvate to pyruvate D with the help of the pyruvate dehydrogenase, which forms the acetyl coenzyme A. Here we have 6 molecules of ATP which is formed. That is conversion of 2 NAD plus to 2 NADH. 1 NADH is equal to 3 molecules of ATP. Here we have 2 molecules of NADH, so we have got 6 molecules of ATP. And finally, 2 molecules of acetyl coenzyme A into the TCA cycle, which produces 24 molecules of ATP. So overall, in the aerobic respiration, the complete conversion of glucose into carbon dioxide and the reactions, we have got 38 ATP molecules, which is there are some important points to be remembered in the TCA cycle. Number one, it is a series of chemical reactions used by all aerobic organisms to release the stored energy through oxidation of acetyl-CoA. And this acetyl coenzyme A molecule is derived from carbohydrate, fats and proteins. It provides precursor for certain amino acids, and we have seen the reactions in the amphibolic pathway. And it provides energy, and these electrons from NADH, FADH2 and ATP is passed to the electron transport chain for further production of ATP. And hope you have learned many points from this particular video, and this will help you to learn the TCA cycle in a better way. Thank you.